Hello, and welcome to part 2 of Adaptive Immunity, where we're going to take a look at the lymphatic system. Now, the lymphatic system is composed primarily of lymphatic vessels and lymphoid organs. There are, of course, lymph nodes as well, and the role of the lymphatic system is really in filtration and cleaning. The lymphatic system works in conjunction with the circulatory system, and it does kind of mirror with the circulatory system. The lymphatic vessels are responsible for carrying fluid from the circulatory system through the lymphatic system into lymph nodes for filtration and presentation to macrophages, B cells, and T cells in that case. The fluid that's found in the lymphatic system is referred to as lymph. And again, it is filtered through those lymph nodes so that the immune system can kind of sample what's going on out in the rest of the body. So it plays a large role in cleaning of the bloodstream, of the circulatory system, as well as filtration and fighting infection. So it's a very important part of the immune system. The secondary lymphoid organs consist mainly of the lymph nodes. We also have the thymus, the spleen, tonsils are even considered part of the lymphatic system. And these are areas where T cells and B cells, as well as natural killer cells, are found in high concentrations, usually mostly T cells and B cells, and this is where the real filtration is going to be occurring. The secondary lymphoid organs are where antigens are encountered by the adaptive immune system. This is also the only place where activation of the adaptive immune system will occur. Now, the lymphatic system, again, will filter blood from the lymph capillaries. Here we can see the circulatory capillaries. We have the venous system over here and the arterial system. And as blood moves through these capillaries and we have the exchange of gases and oxygen and nutrients out into the interstitial fluid, at the same time, the pressure that is pushing that fluid out of the circulatory system will cause some of that fluid to be forced into the lymphatic vessels at the lymph capillary. These small vessels that you can see are intertwined with our venous capillaries. And this is flap-like structures that cause them, they're trying to show you one here, those structures are going to cause the lymphatic system to be a one-way flow of lymph so that the lymph does not flow back out. So here's just a quick video that will do a much better job at explaining the lymphatic system than I am, but to give you an idea of how complex it is and really quite fascinating.
The lymphatic system is a multifunctional body system. Its roles include transportation of lymph and combating infection. The components of the lymphatic system include lymphatic vessels, lymph nodes, and lymphatic organs. Lymphatic vessels form an extensive network of thin-walled vessels that transport lymph around the body, eventually emptying it into the venous system. Lymphatic vessels contain numerous valves and have a beaded appearance, bulging between adjacent valves when filled with lymph. Lymph nodes are small masses of lymphatic tissues situated along the course of the superficial and deep lymphatic vessels. Their function is to filter lymph as it passes through the lymphatic system en route to the venous system. Lymphatic organs are structures that produce lymphocytes and include the spleen and the thymus gland. Lymphocytes are white blood cells which circulate in the bloodstream and lymphatic system and are concentrated in lymph nodes. They are components of the immune system and have a role in combating infection.
So in addition, another part of the lymphatic system that you don't hear about as much is called malt and salt. And malt is mucosal associated lymphatic tissue. So we have in the mucous membranes, mucosal associated. and salt is skin associated. And both of these are layers or they're linings of the body. We have skin, which of course is on the outside of our body. And then we have in mucosal membranes, these are usually internal. And specifically, malt is found primarily in the GI tract. It is found in both the small and large intestine, but mostly in the small intestine. Down here, this may look familiar from your anatomy classes. We have our Peyer's patches right in here. There's some granular tissue in here. Lots and lots of dendritic cells and macrophages, B cells and T cells found in here. The entryway into a lymph node here is called an M cell. M stands for microfold. They're showing you the M cell right here. So these M cells are just a little slightly bit different than the ciliated endothelial cells that are lining the gastrointestinal tract. You can see there's an M cell here and of course an M cell here. And the M cells are intended for antigens that are floating around in the mucosal lining in the gastrointestinal tract to come through and enter into the Peyer's patches themselves. These Peyer patches are filled with T cells and B cells and dendritic cells. And these dendritic cells will consume and sample. The whole idea of a Peyer's patch is kind of to work like a lymph node and to really sample what's going on in that gastrointestinal tract and to tell the rest of the immune system who to react against and who not, who's safe and who isn't. So there's still a lot to be learned about the mucosal associated lymphatic system. There's a lot of regulatory practices that take place or regulatory actions, activities that take place in malt tissue. Once a B cell or a T cell has been activated within the mucosal associated lymphatic system or tissue, it cannot leave. It can't move out into circulation. Now, B cells will be initiated to produce an antibody, and B cells in malt produce IgA antibodies. That's what you're seeing here. So IgA antibodies are what are produced primarily in mucous membranes. Over here is skin-associated lymphatic tissue. I didn't quite finish writing that out for you. So skin-associated lymphatic tissue, and its primary cells in function are the Langerhans cells. And these Langerhans cells will antigen present and begin presenting on their MHC class II. We'll talk about those a little bit. But they also have toll-like receptors, which we saw in the innate system. So TLRs will get initiated. Langerhans cells will phagocytize, a form of dendritic and macrophage. You can see we have all dendrocytes here, dendritic cells here. These are specialized dendritic cells in skin. We also have, interferon 1, tumor necrosis factor. You can see a lot of the cytokines that we have talked about. These are more pattern recognition type receptors similar to it. So we have lots of different, here's a CD4 T lymphocytes. So we have lots of different components that we have already mentioned in both innate and adaptive immunity that are part of this Langerhans cell process in skin-associated lymphatic tissue. So in the lymphatic system, we have the primary lymphoid organs, which are the bone marrow and the thymus. Secondary lymphoid organs include the spleen and tonsils. Some argue that the spleen is a primary lymphoid organ, so it can sometimes go either way. Now, bone marrow in particular is responsible for blood cell production. It is also where B cells will mature to become naive B cells. The thymus is where T cells go for a process known as maturation and priming. We'll talk about priming in a little bit. And T cells, they make that pit stop. Both of these cells, both T cells and B cells, are going to end up in a lymph node as what are known as naive cells. Right? So they will be naive, both of them. And naive just means that they're mature. They're able to carry out a function, but they have not been activated yet to carry out that actual function, to do it. Now, the process of priming is very important in T cells, just to kind of sidetrack here real quick. And the importance of priming is because T cells have to be told what is host and what is not. So a T cell precursor, I just want to walk you through these diagrams here at the bottom. Here, the T cell precursor molecule forms its T cell receptor, and it carries out this process here in the thymus. Once it leaves the thymus, the immature T cell, it will move into the lymphatic system, and it recognizes inside of the, I'm sorry, it's still in the thymus. Inside of the thymus is where it goes through this process of priming. And this is known as positive and negative selection. Now, what's really happening here in the thymus is the the T cell is being presented all kinds of different host antigens. So anything that is part of our own cells, our own cellular receptors and proteins and parts and all of that stuff, are all being presented to this T cell in multiple ways in this very small area of the thymus. And if the T cell reacts with any of those host antigens, any of those host markers, the T cell will be removed. And so it will be destroyed and moved into apoptosis so that this T cell cannot activate any part of the immune system against the host itself. Once this cell passes the test in the thymus to become a full-blown T cell, it is now a naive T cell and will sit in the peripheral lymphoid organs, so it'll be in a lymph node. And when it's in that lymph node, it may come across antigen presentation. And so antigen presentation will occur, and this will cause the T cell to then proliferate and start activating macrophages as T helper 1s, or start activating B cells as T helper 2s, and or becoming a cytotoxic T cell and going out and telling virus-infected cells to go into apoptosis. Over here on the left are the B cells, the process of B cells. And the B cell precursor rearranges its immunoglobulin genes. It predetermines what it's specific for, you could say, when it's in the bone marrow. Once it matures enough to leave the bone marrow, it will have an IgM antibody bound to its surface, and it will look at parts of the bone marrow to see if it reacts with any self-antigens. If any of those receptors, B cell receptors, react with any self-antigens, that B cell will be removed right then and there in the bone marrow. Once it passes the bone marrow test, it now matures and it moves out into the lymphatic system. If the B cell binds directly to an antigen itself that is known as a T-independent antigen, then the B cell can be activated. If it needs T cell to present an antigen, then that would be called a T-dependent antigen. Here they're showing the co-reception of IgMs. This is an IgM here. And then also regulating all of these processes are the IgD antibodies. IgDs are considered co-receptors. Once a B cell is activated, it's going to become one of two types of cells. It'll become either a plasma cell or a memory cell. Plasma cells are responsible for secreting antibodies that will be released into circulation, and memory cells will have IgG antibodies on their surface, and they will stick around for months and years, and they are sticking around waiting to see if that pathogen ever comes back. So that's the primary life cycle, you could say, of B cells and T cells. So just to review what's happening in the lymphatic system, what is, of course, the primary function of it? What are the three primary structures of the lymphatic system? Where are B cells and T cells found in the lymphatic system? What are secondary lymphoid organs? What is MALT and where is it found? And of course, what is SALT and where is it found? Just a quick note here, these pictures right here that I put in the small circles, that is SALT tissue. That is fish microscopy or fluorescent in situ hybridization. And they use the fish microscopy method to fluorescent stain actual salt tissue in a skin, in a layer of skin. Just interesting little picture there. So that's the end of part 2 on lymphatic system within the adaptive immune response. I will see you all in part 3.